episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome everybody again to another episode of our show with another fascinating guest involved in creating a better tomorrow for all of us. And today we are going to be continuing what I've been referring to as our virtual road trip and heading around the world to Singapore to visit with none other than Dr. Brian Kennedy, who is a distinguished professor in the Department of Biochemistry and Physiology, Yanglu Lin School of Medicine, National University of Singapore, Director of the National University Health System Center for Healthy Aging, Professor at the Buck Institute for Research on Aging, Adjunct Professor, Leonard Davis School of Gerontology at USC, and affiliate faculty, Department of Biochemistry at University of Washington. With his Ph.D. in biology, Dr. Kennedy from the Massachusetts Institute of Technology, MIT, has a pretty broad focus directed at understanding the biology of aging and ultimately translating various research discoveries into a variety of ways of delaying, detecting, preventing, and treating human aging and associated diseases. Dr. Kennedy's current research projects involve many things involving the systems biology strategies to understand aging and a variety of murine longevity studies and disease models, which he conducts in his lab. Dr. Kennedy has published in over 80 manuscripts in prestigious journals, including Cell, Nature Science, Genes and Development, preceding National Academy of Sciences, and currently serves as co-editor-in-chief of Aging Cell. Dr. Brian Kennedy, thanks so much for taking the time to come on the show today.
Oh, it's my pleasure. Thanks for having me on the show.
Most definitely. Brian, there's not too many people in sort of the biotech world that don't know who you are, but for some of our viewers that are sort of outside of this space, I'd love to just start off, as we typically do, by handing you the floor for a few minutes just to talk a little bit about yourself. If you can sort of take us back to the beginning, everything from where you grew up to how you developed your interest in biology, and ultimately what put you on this journey into the field of aging and healthy longevity. I think that'd be a great way to start everything off that we're going to be talking about.
Thanks. I grew up I was born in Louisville, Kentucky, and grew up in suburban Louisville. And I've been gone long enough that I've lost my Southern accent, but I promise you that's where I come from. My parents were both teachers in math and chemistry. And so I guess it was ordained that I was going to head down a science path. And I went to Northwestern, but then when I went to graduate school, I started talking to Lenny Guarente, who was a well-known person in the aging field, as I'm sure you know. At the time, he wasn't working on aging. He decided that he wanted to do something different in yeast cells. And there was another graduate student, Nick Austriaco, who's now become a priest, by the way. So that's an interesting discussion. He and Nick and I thought about what we might want to do in yeast cells. These are single-celled organisms. And so we thought that, the two craziest things we could think of were programmed cell death and aging. And we thought that programmed cell death in a single-celled organism might be a little too crazy. So instead, we started studying aging, and that was the beginning of it all. It turns out there's a whole field now in programmed cell death in yeast, too. So I guess we would have been okay either way. So that, those early days, that's where we found the mutations that were in the sirtuins in yeast. And that started the whole sirtuin aging craze. Ran away from aging, did a postdoc at Harvard Medical School on tumor suppression. But when I came back to the University of Washington as an assistant professor, I was really still enamored by studying aging. And the thing I liked about it was that we just knew so little. It was like an open field, open avenue to head down to understand biology. I think even at that time, I still wasn't really thinking about slowing human aging. It was more about how do we understand this process. And so we went back to yeast cells, and then we screened the whole genome for mutations that make yeast live longer. Started working with Matt Kaberlein, who I'm sure you also know. He also was in Lenny Guarente's lab at MIT. Mm-hmm. We hit on the TOR pathway and a bunch of other pathways too. And so that was one of the papers that really put TOR to the forefront of aging. And so eventually I decided that I have to get out of yeast at some point, although we still dabble in it. And so we got into worms and mostly mouse studies in aging. And a lot of my lab still works on that. But now we've stopped, we're not only poking and prodding mice, but we're poking and prodding humans over here in Singapore. Because we think it's time to really take these interventions that have been developed and test them and validate them in human clinical studies, because I think we're not that far from interventions that can really slow or even reverse aspects of aging, and it's time to find out what works and what doesn't.
Absolutely, absolutely. And Brian, looking through sort of the work that your lab currently does, and these two fascinating areas, one that I'd love to start off on, and that's just sort of the principle of the systems biology of aging. And I got about a year ago, I got to spend about 90 minutes with Lee Hood over at the Institute for Systems Biology, sort of the father of this space. And I heard you talk about sort of the systems biology of aging and sort of this concept of the preservation of the homeostatic networks, these networks that we don't normally think too much about when we think in the traditional pharma sense of looking at a specific target or whatnot against a specific disease. But a lot of the ways these very complex mechanisms, some that you just mentioned in terms of TOR and so forth, really aren't isolated by themselves, but interact in really fantastic ways in all sorts of directions. Talk a little bit about some of the things that you look at in terms of the systems biology of aging, and what some of the areas you think maybe are a little underexplored in regard to systems biology.
Well, I think you've touched upon really, I think, one of the conundrums still in the field. So, about 7 or 8 years ago, there were 2 papers published, one on hallmarks of aging and one on pillars of aging. They're both reviews. I was involved in one of them, and we more or less, there were a large number of professors on both papers. It was kind of a consensus in the field that there are certain pathways that modulate aging, and these are things like adult stem cell function. The pillars and hallmarks don't completely overalign, but they kind of do, in inflammation, cellular damage, that sort of thing. And so then the next thing you can do is you can say that we have some small molecules that target pathways that we know extend lifespan and health span as well in animals. So you might assume that they hit one of those pathways. And so people have been looking at each pathway of aging and to say which one does rapamycin hit. Now we know rapamycin inhibits the TOR pathway, right? That's the direct target. But the question is, what's downstream of that? Is it reduced damage, better repair of damage, reduced inflammation, et cetera, et cetera? And the problem is you can read out the benefits of inhibiting TOR in all of these pathways. And that's true with sirtuins. It's true with AKG, I think, which we're getting excited about. And so what does that mean? Does that mean you have molecules that magically touch everything related to aging? It seems unlikely. And so that led me to think about this idea that, we can call things different pillars of aging, but really they're connected to each other. And so, if you have altered metabolism, that affects inflammation. And if you have inflammation, that causes cells to senesce. And if you have, and that causes damage to cells. And so, you can tie everything together in a network. And so that's how I think of it, that you have this network that preserves homeostasis in the body. And so things are going wrong as we get older, but we're still highly functional because this network compensates for the damage that's happening. It keeps us active, it keeps us disease free. But when enough aspects of that network fall apart, then we start to have chronic diseases. So I think that the way I think about it is that the TOR pathway is at one of these nodes in the network. So you can read out the inhibition of TOR in all the different pillars that are on the edges of the network. But the truth is that the things that are really affecting aging are involved in network preservation. And I think that it's easy to say that, but it's hard to understand what that means. And so we're grappling with that concept now. What does it mean to be, what is this network? What does it mean to be a node in this network, and how do we define that?
And you obviously you spend a lot of time over the years, working with yeast, working with the mouse models. You published a lot with you say Matt Kaberlein, and who's active with dogs. Andrei Gudkov also in the space. I spent a lot of time sort of going through your publications, and I was fascinated by one that popped up recently called Nontraditional Models: The World Goes Bats, Living Longer and Tolerating Viruses, which you did work with Vera Gorbanova on this one. I had her on a couple of months ago, of course, talking about naked mole rats. I also had Shelley Buffenstein from Calico on, who's also a naked mole rat person. I'm interested in, obviously we got the mice and the dogs and everything fine. Humans fine, and we're working in that direction. But there's a lot of other stuff there. And Shelley Buffenstein from Calico is very fond of saying, look, there's a lot of other animals that live really long that we should be studying and learning from, in addition to the naked mole rat. Talk a little bit about stuff that you're doing outside of the traditional animal models, because some of these species, like the bats and their heat shock protein, defense, and all the stuff that makes them so tolerated to viruses, which helps with their longevity as well. Talk a little about this, if you would, and some of the things you're interested in studying there.
Yeah, it's fascinating, these bats. I think that everybody kind of is paying attention to them now, and they're endemic to everywhere in the world, really. But there are many different species here in Southeast Asia, so there's a lot of interest in the bats here, and of course they're carriers for all kinds of disease. And so the truth of the story is that Vera and her husband Andrei Selovanov, they're visiting professors here in the U.S. And so they were visiting us when the pandemic really hit in Malaysia and Singapore, and everybody got nervous. They were actually we were all taking a weekend at a resort in Malaysia, and Singapore said, you better get back to Singapore or you may not get back. So we all came back, and they were planning to spend an extra few days in Malaysia, and they had to change their plans, and they were kind of just stuck in lockdown in my apartment. And so we thought, how can we make use of this time? And so then we came up with the idea of writing a review on bats. Now, Vera does a lot more work on that than I do, but we have collaborators here that have bat colonies as well. And so we're very interested in this. These bats live a very long time, some of them up to 40 years. And they're amazing species. I go for runs at night in Singapore, and there are bats all over the place. If you go to the right place, you can see these fruit bats that are just huge, flying right above your head. So they have very high metabolism. They have a very interesting lifestyle. You wouldn't look at these things and say this is something that's going to live a long time, and yet they do. And it really, I think, ties in somewhat to senescence, because they have alterations in their innate immune system. And so their cells, when they get cytoplasmic DNA, which happens or cytoplasmic nucleic acids, which happens in virus infections a lot, our cells go crazy, evoke innate immunity. You can get cytokine storms and all kinds of problems as a result of that. Their cells just tolerate it. So they have mutations that allow them to tolerate these viruses. And so they may live for 20 years with the coronavirus, and so and we think there's some kind of link between these changes in innate immunity and the ability to live a long time. Also, bats also have metabolic changes that are may allow them to live a long time too. So we want to start to interrogate those things and figure out which ones are most relevant to aging and how that relates to innate immunity. And
Brian, somebody else that I had the opportunity to spend time with about a year ago, Dr. Victor Zhao of the National Academy of Medicine, talking about the Healthy Longevity global competition. I noticed that you and your team were an awardee recently. The title of the work that you're going to be doing under the NAM program, studying epigenetic aging and telomere length as possible biologic predictors for healthy aging. And it's then sort of the subline is the Singapore Chinese health study. I was wondering if you could just talk a little bit about this model. You know, obviously we have these interesting new models nowadays, and the healthy longevity competition is one of them. You know, I'd love just to talk about how you're looking at some of these new models, like the NAM model, and sort of funding some of this earlier research that's not sort of typically funded by your VCs or for other types of investors. And then if you could also just talk a little bit about some of the, because it looks like you're looking at sort of in this case, sort of the biomarkers, sort of systems biomarkers, not just one thing, but how a lot of these things interact, alike with your network approach to aging. Talk a little bit about what you're going to be looking at specifically in terms of how these different biomarkers sort of interact together in this population.
Yeah, so first of all, I think the National Academy of Medicine grants are great, and they're in the United States originally, but several other countries have picked up on this, including Singapore. And so there were, I think, 13 awards last year for grants, and a lot of them come from our Healthy Longevity program here in the US, so I know about a number of them. So we're excited about that. And it's kind of a pyramid structure. So you get a small grant to start, and you do some initial pilot work, and then you apply for a second round of grants that are $500,000 in the US. And then there'll be an award, or at least one award, of a $5 million grant at the end of this if the intermediate studies go well. So it's a different kind of structure for funding, but I think it's exciting. And I've been involved in the review also in the American grants, and so I've seen a lot of the things that have come through, and there's some really innovative ideas. The goal is to really have ideas that are not easily fundable through the NIH because they may be earlier stage or more risky, but also to develop things that have value for aging people in the near future. And so there are things that are funded on all aspects of aging, not just on altering the biology of aging. So I think that it's part of a movement that's really led to funding of aging research in the last 5 to 10 years, and I think that's really exciting. So private sector money's gone way up, as you know, and there are now different mechanisms for public sector funding as well. So it's really, finally, after years of begging for money, I think the aging field has a little bit more access to money, although I would still argue it's heavily underfunded from the government side. So what we're trying to do over here, that grant is part of a big picture of looking at a whole range of different biomarkers of aging together in the same people in Singapore, because we all think about interventions to slow aging as the great breakthroughs in the aging field, and that's certainly one area where great breakthroughs have been made. But we should also consider these new biomarkers as great breakthroughs, because before that, there was really no way in a non-frail person, there was really no way to effectively measure biologic aging. And now we have a number of candidate approaches to do that. DNA methylation is something you've probably talked about on previous shows. That was developed in large part by Steve Horvath. Now there are many different methylation clocks that predict biologic age. That's probably the leading example right now, but we're also using more standard assays of pathways like telomere length or TOR signaling. We're looking at inflammation. We're looking at, we can recreate a biologic age from facial patterns. So we're working with Jackie Han at Peking University on that. We're working with a company called Gero to look at some of their biomarkers of aging. So what we want to do is to measure a lot of different biomarkers in the same people, figure out how much these different markers overlap with each other, and how do we put them together to get a holistic picture of a person's aging. And ideally, how do we do it for a relatively low cost? Because I would like to scale this into the Singapore population and stratify the rate of aging of everyone when they hit, say, a particular age, 40, 45, 50, something like that. And then the other value to this is that these biomarkers can be used as endpoints for the interventions. So when we do a study, clinical study with something like AKG or an NAD precursor or exercise, we can use these biomarkers to see if we're having an impact. And hopefully, these interventions will actually reverse biologic age using these biomarkers. And there are a few publications which suggest that might be possible, but it's still very, very early days. So one of the really exciting things in the field now is to putting the interventions together with the markers of aging and seeing how they interact with each other.
And, you know, this is going to be, as you were saying, just really critical, especially in sort of the younger, non-frail population. You've been very successful in pushing, as you were saying earlier on, some of these initial human experiences. And I'd love to get sort of your thoughts on, because you're in a part of the world right now where there's been, let's say, a little more creativity with regard to accelerating some of these novel therapies. Obviously, we think of conditional stem cell approvals in Japan. China has been having some interesting sort of free zones for sort of experimental therapies to move a little quicker. And then, you know, I have some, you know, still have some friends in the pharmaceutical industry and in certain companies where they have these weird sort of phase 0 clinical study groups where they once again look at interesting ways to, you know, to test an initial population of 10 people here and 15 people here. You're seeing a lot of interesting movement on the clinical development front. In other areas, and then, you know, obviously think about how some of these tools you were just mentioning before can help sort of balance and sort of give more confidence within some of these novel models that, hey, things are happening in this sort of early phase 0/1 period of time.
Yeah, you know, it's things are changing. I mean, I've been talking to pharmaceutical companies for 15 years about targeting aging, and there are two problems with this. One, as you know, that the FDA doesn't recognize it as a disease indication. And so, you know, a company like a pharmaceutical, well, a big pharma, I won't mention any specific ones, a big pharmaceutical company, you know, the bottom line is what matters to them. And they have a tried and true method of making money. And that is to develop therapies for disease indications and get reimbursement for treating sick people. Now, there's nothing wrong with that. I want to treat sick people too. But the idea of preventing aging is complicated for them because it doesn't fit their reimbursement model. And it doesn't, it also could take away from their therapies if we're keeping people healthy. You know, the complicated cancer therapies are not going to have as much value. So they haven't led the way in the aging field like you would hope. And I think just now, in the last few years, they're starting to look into this, but they're still not leading the way. You know, what's happening is that small biotech companies are getting started. We started several of them at the Buck Institute, and we're doing that over here in Singapore as well. And a lot of other people are as well. And so now you have probably 50 or more different small biotech companies thinking about aging. Some of those are going to be successful, start to have really, really great preclinical data, get to the clinic, maybe with a disease indication or maybe looking at aging directly. And eventually pharma will swallow them up and, you know, take it to the end of the game, I think. But right now, that's where the innovation is happening, not just in academic labs, but in these small companies. And then on the other side, you have things like stem cell replacement therapy, which I'm very interested in right now. We're starting to think about how to look at people that get mesenchymal stem cell by IV and see if that has an impact on aging. But that, you know, was slowed down dramatically when the Bush administration decided stem cells were persona non grata in the US. And of course, now people are using iPS cells or MSCs derived from umbilical cord or things like that. And it's mostly not embryonic stem cells, at least in the aging field. But, you know, what happened is all these clinics developed outside the US, and we never had the really good placebo-controlled clinical studies to figure out what the mesenchymal stem cells do when they're put in your body. So I think there's a lot of promise in regenerative medicine, and we want to sort of go back now to try to evaluate, what are the benefits of having mesenchymal stem cells derived from umbilical cord or from your own fat or from wherever put back in your body. How does that impact aging? And there are other areas too that I think that need research, like natural products we've gotten into in the last few years, because it's the Wild West. When you go into a GNC store and there's lots of things that say anti-aging, I really don't know what works and what doesn't. I don't know what's in the bottle half the time. And so the sort of basic biology of aging field that I've been in has typically said, oh, it's all bad science, we're not going to pay attention to that. But I think that's also the wrong approach. I think some of these things are going to work. But we have to go back and do the preclinical research and the clinical research to validate what's working and what's not. So what we're trying to do now is to be, especially on the academic side, is be as agnostic as possible. Let's look at many different kinds of interventions, compare them to biomarkers, and in some cases to disease onset, and see, what works in what context in which people. And I think that's the next step toward getting widespread adoption of longevity-based therapies.
Yeah, completely agree with you on that. Brian, hitting on something you just mentioned before, and that was the topic of sort of the FDA not recognizing aging as a disease yet. Obviously, there's initiatives like the TAME trial and so forth that's trying to shift things. I was just, the question I always have about this one, I don't have an opinion one way or another, but thinking back to my time. So I was at the FDA a few times in my career, and I spent some time looking at some of the drugs that were recently approved in the last year. And you look at the indications, and it's not like here's one. This is for a Sanofi Aventis product called Sarclisa, which is for multiple myeloma. So the indication that they got from the FDA to be used in combination with carfilzomib and dexamethasone for the treatment of adult patients with relapsed or refractory multiple myeloma who have received 1 to 3 prior lines of therapy that did not work or are no longer working. And I say to myself, the FDA, even if they ultimately approve, come around, I worry in some ways that aging might get fractionated in a strange way in an indication. And I think back to sort of, remember those original Cheerios sort of claims, like originally Cheerios are good for your cholesterol, but now the claim is, Cheerios, if you eat 4 bowls and you eat a healthy diet, and it might be good for your cholesterol. What do you think is going to happen ultimately if FDA or EMEA or whatever actually comes up with an aging indication? What do you think it's going to look like?
Yeah, I don't know. And I have some of your same concerns. And I'm very supportive of the TAME trial with metformin also, but it's a huge trial. It costs tens of millions of dollars. And I don't know if metformin is better or worse than rapamycin or AKG or anything else. And what we'd like to know is, how do they compare to each other? And that's why we chose to go after shorter term studies with biomarkers here in Singapore. Also, it's just not a big enough country to do a trial with 3,000 people at 14 different sites there. That would, Singapore can't do that. So I think that doing these kinds of health span and disease prevention trials are interesting, and that's one path toward getting widespread adoption, but it's not the only one. And I also would comment on the FDA. The FDA is not there to innovate.
Right.
What we're talking about here is a medical revolution. We're talking about doing healthcare for a change and not sick care. And that has all kinds of connotations, both in the healthcare world and in the world at large. And so I think that you can talk to the FDA, but you can't expect them to solve your problems for you. I think what we have to do is create problems for them. And that's doing clinical studies that show that this approach is valid, that it works, and then combining with the economists to prove that this has much more economic benefit than treating, diseases one at a time. and then forcing them to take notice and react to it. I think that's how you get regulatory change. You don't, regulators don't make the change, they get
No.
forced to make the change.
Right.
So that's our approach. This create problems for regulators and make them solve them. And I agree with you that the system right now is not set up to deal with something as big as aging. And it could be, and if they're saying, well, we approve this in longevity medicine for people that are rapidly aging in the Delaware climate, it's not going to solve the problems we're looking for. And having said that, I don't think any intervention is going to work in every individual, and we do have to understand which type of people react to which longevity interventions, and so that personalization is important to head toward. But let's take even a step back. Every time I walk through NUS, there's a big copy on, a sculpture of the Hippocratic Oath, right? So and when you think of Hippocratic Oath, you hear first do no harm, right?
Mm-hmm.
So this is, well, first of all, it was a revolutionary idea of its time. And I still agree with the concept, but I think we misinterpret the concept. We interpret the concept to be wait till somebody gets sick and then treat them in ways that are, clinically validated to help them. And so the good outweighs the bad. And we also interpret that to mean that if somebody's healthy, you shouldn't do anything to them.
Yeah.
And I think that second component is wrong. I think medicine and healthcare should be a lifelong process. And just because somebody's 40 and they don't have any clinically defined condition doesn't mean that we shouldn't be thinking of interventions and strategies to make them biologically 35, to make them healthy much longer in their life so they don't get sick later, to treat them now when it's easier to do it than it is to wait till they have Alzheimer's disease, and it's much more difficult to do it. So I think by not helping people at certain stages of life, you are harming them. And we need to really think about what that oath means and whether we shouldn't be working with people on a much more consistent basis before they get sick to try to keep them healthy. That to me is what do no harm means.
Right. You've mentioned AKG or alpha ketoglutarate a bit today. I know this has been not just an interesting research program of yours, but also some commercial initiatives. Talk a little bit about what AKG is for those that might be unfamiliar with it, and how you got interested in it, because this is a natural product, right?
Yeah, it's your body makes tons of it. It's in the TCA cycle, and so it's in central metabolism. The problem is you have less of it as you get older. So in some ways it parallels the NAD story, which I guess you probably talked about in previous videos, right? This is another metabolite that goes down with age, and when you supplement it back up, it seems to have some benefits. The difference is here we get a really big impact on compression of morbidity with AKG. So when we treat animals, they live about 10% longer, but they're healthy, and that's judged by this mouse frailty index, much longer. So you get animals that are doing really, really well and then drop off at the end very quickly. That's a much better way to go than to have debilitating diseases for 20 years in humans. And so, of course, we're doing clinical studies now in humans to see if that translates. But coming back to AKG, it does a lot of, it probably participates in about 1,000 different reactions in cells. So it's a little bit of a challenge to figure out which ones are important. But we found that when we give it to animals orally, that they, their adult stem cells function better, their metabolism's better, they have better skin and coat condition, they don't lose their hair, for instance, they don't get kyphosis as badly. So there's a whole range of aging parameters that are delayed with AKG. So this is a very safe reagent. It's been given at very high doses in clinical studies for other purposes, not for aging, and with no adverse events or side effects. So we're very excited about it. A company called Ponce de Leon Health funded Gordon Lithgow in my lab at the Buck to do these studies, and we looked at a lot of different natural products, first in worms and then in mice. And AKG is the one that really jumped out at us as having a big effect. It was reported in Nature by another group in around 2014, I think, showing that it had a big impact on aging in worms. But there's a lot of things that are reported in animals to have effects on aging, and they're not so easy to repeat. AKG was really easy for Gordon to repeat in worms, and then we moved to the mice and found, these exciting results as well. So now we're gearing up to, the company's doing clinical studies with their product, which is Rejuvent, but that's sustained release alpha-ketoglutarate plus a vitamin that in our studies in mice had synergistic effects on frailty. But what we want to do over in Singapore is to work academically just with the sustained release AKG and try to get a mechanistic understanding of what it's doing to human aging. And so we'll hopefully be starting those studies soon over here. There are lots of different candidate mechanisms for why AKG might affect aging, and we want to systematically go through those and try to sort out which ones are most important.
Very nice. Brian, another fascinating paper of yours that stood out to me, and I think this goes to sort of your profile as being looking at this whole area very holistically. Was a paper entitled "Effects of Choral Singing versus Health Education on Cognitive Decline in Aging," randomized controlled trial. Obviously, there's sort of the segment of the longevity biotech world that thinks all we're going to need is some pills and stem cells, and there's some other folks that think, nah, there's a much more integrated set of stuff we're going to need to do. Talk about what you learned in this particular study and why you looked at it.
Yeah, well, I'm not the principal investigator on that study. I helped out with it over in Singapore, but I'm happy to talk about it. And I think that the conclusion that people are going to get from listening to this podcast is that I have attention deficit disorder, which may actually be true. I don't know. But one of the things I'm over here to do is to enable scientists to study aging. So if people come with good ideas, we want to help them out and help them with design of studies, help them with endpoints. And look, lifestyle interventions work. I mean, I'm more into exercise and diet in terms of research and life than I really can't stand choral singing, but to be honest, full disclosure. But there's another thing that's really known to impact aging, and that's social connectivity. Right. And so if you bring, and this is not a study in 50-year-olds, this is a study in older people that have early stage dementia or cognitive, mild cognitive impairment. And the idea is to bring them together, have them participate in singing, which is also on its own has been shown to have some benefits, and also get them connected and interacting and see what the impact that has on later stages of aging. So, there's preventing aging and managing aging. To me, this is more on the management side. But that's still important. You need to improve life quality for people, and increasing their connectivity, having them participate in these activities seems to be beneficial. So I don't think we're going to live forever by choral singing. In fact, it might drive me crazy long before that. Personally, I wanted to do like rock music, but I got outvoted. I think it does help people, these kinds of lifestyle modifications and interventions and participation in these things. A lot of people in Asia, when they get older, they live with their parent or their kids, and they become very isolated, particularly if they can't get out of the house and get around. And so anything that gets people moving around and interacting with each other, I think, is good. There's another study on gardening that we're helping out with as well. So again, I have a very broad view on that we need to change all of society to better suit the aging population. And so I'm interested in any way you can do that. I mean, most of my direct research is on small molecules of drugs. And I would much rather prefer just not to get dementia than to have to manage it. But at the same time, we have a dementia pandemic going on as well. And so we need to do something to help people at all stages of aging.
Yeah, I was. That's where I was going with that one, and I think it's just a side note. It's extremely important, and not just for dementia, but just all sort of the whole area of delirium as well, which is sort of dementia's evil cousin, and
Yeah.
some of these we don't normally think about, but yeah.
Well, we're doing a study kind of on that too. That post-surgical delirium
Yep.
is a major complication for the aging population. And so
Yep.
one of the great things here at NUS is we have a great hospital, great clinic, clinicians who are interested in doing science. And so we want to use these aging biomarkers before people go into surgery. We're going to start with elective surgeries because it's more complicated with acute needs. But and we want to see if biologic aging predicts surgical outcome, risk of delirium post-surgery, recovery time.
Yep.
If somebody's getting knee replacement, there's some people get knee replacement and they go through the rehab, everything works, and they go back to their relatively normal lives. And other people really never get that mobility back after knee replacement, and it's hard to predict who's going to respond and who isn't. So these biologic aging markers may have value in those contexts as well.
Yep.
We can assess people and say, all right, based on your biologic age, you're 8 years younger than your chronologic age. If we do this knee replacement, we think you're going to do very well. I mean, that's a hypothesis. We don't know if it'll work or not, but we're trying that concept.
So, Brian, if someone held a gun to my head and I had to pick my favorite Brian Kennedy paper, the one that I have to select, and I'll tell you why.
You're going to pick one in a minute that I can't remember, so be careful.
No, you'll remember this one. And this one I really enjoy, just for a variety of reasons. We can get it offline, but spatiotemporal correlates of gene expression and cortical morphology across lifespan and aging. And in this paper with you and your co-authors, you go into the developmental theory of aging and basically, looking at aging as sort of this reverse process of development. And, I always, I get into this discussion, a lot of people are unaware of the fact that, as we are developing in our mother's wombs, our brains, full of beta amyloid and tau and LN1 and so the whole, you can look at the fetal brain and it looks, just as diseased as the elderly dementia brain, but we don't develop Alzheimer's and Parkinson's in the womb. Talk a little bit about, in general, sort of the developmental theory of aging. Are you working a lot on that? Obviously, you work on a lot of stuff, but is this a hot area for parts of your team or other areas of the university? Because this one is kind of near and dear to my heart for a variety of reasons. So I'll get into that offline.
There's a professor here named Chi, whose name I never get right, but I do my best. And she's been excited about this. She does a lot of artificial intelligence and bioinformatic analysis. And so she was able to get access to transcriptomics and also images of brains from people that are at all ages of life. And so she's been doing this kind of analysis, and it really reinforces this concept that, aging is like the changes that happen during old age sort of, I'm going to say it the opposite way of you did, which is that the changes that happen in old age sort of recapitulate sort of in reverse developmental processes. And so the transcription profile of really late stage brains have some characteristics that look like juvenile brains. And so you can view that, I think, multiple ways. I mean, one way you can view it is that development is a process of building complexity, and that when that process falls apart in old age, it falls apart and the layers get pulled back off, and you get this is sort of the passive view of it, that the layers get pulled back off, and that that just happens to fall in some path that mimics how they got put back on. So it looks like that you're recapitulating development, or that there's some loss of organization, and that noise is more similar to what's going on developmentally. It may be more interesting than that. It could be that in the aging brain, there are a lot of signals for rejuvenation. There are a lot of you know, the damage that's happened is telling you to, like, you got to go back and rebuild things to fix them. And so the cells are trying to do that, but the capacity is not there. And so you get that sort of developmental profile as well. I think it's a fascinating conceptual thing to think about. This study was done in the brain, but it's not, could be done anywhere in the body. There's really, we're still at this sort of stage where we're identifying this process, and we don't understand mechanistically what it means yet. But these changes that happen with old age, I think, are very fascinating, and the fact that they overlap somewhat with development is exciting as well. You know, even senescence, right? You know, there's a role for cell senescence during development.
Yeah.
And nobody thought about that, you know, until recently.
That's why we don't have frog hands and tails.
Yeah, but regenerating things would be good. So maybe.
Yeah, that's how I was going down that path as well. But yeah, it's if I could bring down $1 trillion from the heavens right now, I'd give it to you and say, put some of it into the DTA. That's it's a fascinating,
This brings up a point that I want to hit on, that I think that is a problem right now, is that things have gotten so translational, you know, and not just in aging. I mean, probably aging is not a good example even, but in disease-based research, things are so translational. Even NIH, when they're really interested in how your project is going to lead to a new therapy.
Yeah.
Of course, we want to help people. I'd like to live longer. I'm not immune to this, but I'm really worried about where funding for basic science is going. And there are big questions in aging that we don't know the answer to. We just raised one of them. You know, it's getting really hard to get funded to do yeast research anymore. And I feel bad, because there are a lot of exciting ideas and young scientists that want to study them, that it's really hard to get funded. And you talk about systems biology, you know, I would argue that a mouse is still a bit complicated for that, because you've got lots of different cell types. They're all talking to each other. It's cell autonomous and cell non-autonomous. If you really wanted to understand aging at a complete level, we should be doing it in yeast cells, because there we have one cell, and we can really interrogate how all these different proteins are talking to each other in ways that you can't do anywhere else. And I really think we're selling ourselves short in the long run if we don't fund that basic science, and it's happening everywhere. But this move toward translation, and there are good elements of it, but we shouldn't run away from the beginning of the pipeline, because that's where the big discoveries happen. That's where we redefine what we are as biologically as human beings, and if we don't put money into that, we're going to stop learning new things.
Yeah, and I completely agree with that. And I had about a couple months ago, I had Mike Levin on from Tufts, who's, you know, he leads the CRY out in the sort of this area of sort of bioelectricity and tissue patterning and complex form, how you know, because form and shape, and you know, this isn't dictated by a specific gene. There's much more sort of complex biophysical stuff going on, and you sort of think back that, you know, 100 years ago, we were talking about this electricity stuff, and it just went away. But now, you know, here we are at 2021, and now hey, it has a place. And we also, you know, I think we forget too much, unfortunately, about what happens in sort of the history of the sciences, especially the biomedical sciences, and we really need to not just focus on everything that's going on, but also on maybe things that were left behind and revisit a lot of it. Natural products, as you were pointing out, extremely important.
And it's also like just stuff that we do every day now and take for granted. The internet the technology to generate the internet didn't come from somebody saying, I want to make an internet, you know. And so asking exciting questions and you know, then following the results can lead to very serendipitous benefits that have huge impacts on humanity. Where would we be in this pandemic if we didn't have the internet? Although Zoom is starting to drive me crazy, to be honest. But still, without it, I did a 2-week quarantine when I came back to Singapore recently in a hotel room. And imagine if you didn't have cell phones and the internet, where you would be. And so these things, there's serendipity behind every discovery. There's people asking basic questions behind every discovery. And sometimes the questions they're asking have nothing to do with the ultimate technology that derives from it. And if we don't let people ask those interesting questions, we're in trouble.
Yeah. Brian, it's been an awesome time talking to you. Any other things that I didn't touch on that you might want to mention? Anyone you want to shout out to? I'll give you the floor on the way out. But if I try to hit on all the exciting stuff, but if there's things I missed, please take the floor for a few minutes and give us a final message, if you would.
Yeah, no, I think we covered most of that. I would, having just talked about basic science, let me come back and end on the translational aspects of aging, because I really think we're at the, people have been thinking about slowing the aging process for thousands of years. And I think this is going to be the generation and maybe even the decade where it happens. And so it's really exciting times in this field. There are more and more people that are investing. There's interest from all aspects of society. The general public is starting to take notice. The government is starting to pay attention, as Singapore is very progressive about tackling its aging problem. So I'm really excited. I think big things are going to happen, and in the not too distant future. And I really hope that we can look back and say that, we're taking interventions that extend our health span. And it's something that almost everybody listening to this show will have access to. So I'm excited about where things are going. And there's a lot of validation, and there's still some hurdles to go clear, I'm sure. But I really feel like we're getting to the point where we can really do some good.
Outstanding message. Really great time talking to you, Brian. For everybody that is either going to be listening to this show on the podcast or watching on the YouTube network, you've been listening to Dr. Brian Kennedy, Distinguished Professor, Department of Biochemistry and Physiology, National University of Singapore, Director of the National University Health System Center for Healthy Aging, Professor of the Buck Institute for Research on Aging, Adjunct Professor, Leonard Davis School of Gerontology at USC, and Affiliate Professor, Department of Biochemistry, University of Washington. Brian, it's really been an honor talking to you. I want to take time to thank you for taking time out of your schedule to come on the show. Thank you for everything you're doing there and around the world. And as we say here on this show, thank you for helping to create a better tomorrow for all of us through the work you're doing. It's very inspirational.
Helping disseminate this knowledge is really important. So I appreciate what you're doing too. Thanks a lot.
Absolutely.